Hello everyone, my name is Joyce Kang, and I will be presenting on how a new metagenomic sequencing approach called read cloud sequencing has allowed us to elucidate microbiome dynamics within the context of patients undergoing hematopoietic cell transplantation. Next-gen sequencing has revolutionized how we study the microbiome via metagenomics, which is the sequencing of an entire community or mixture of organisms directly from a sample, such as a stool sample, often without a priori knowledge about which organisms that sample contains. One key challenge within metagenomics is that conventional short-read sequencing and assembly approaches have difficulty resolving genomic regions that are longer than the sequenced fragment length, often 50 to 300 base pairs, and that are present in multiple copies within the metagenome. Some biological examples of repetitive sequences like this are insertion sequences and antibiotic resistance genes present in multiple copies within a metagenome. Our lab has developed a new approach called metagenomic read cloud sequencing, as well as a custom assembler called Athena that helps to overcome some of these challenges. So, how does read cloud sequencing help us with assembly? Well, if we look at this example, we have two instances of a repetitive region called R, once that's located between sequences A and B, and once that is located between sequences C and D in two different spots in a genome. If we use conventional sequencing, such as an Illumina TrueSeq library preparation and a conventional short-read assembler, such as MegaHit, then we'll be able to get back sequences A, B, C, D, and R, but we won't know whether A goes with D or B, and we won't know whether C goes with D or B, because there's an ambiguity since R is repeated and the sequencing reads are shorter than the length of R. Meanwhile, read cloud sequencing, also known as linked read sequencing, is a relatively new technology that allows us to get around this problem. The main difference is during the library preparation stage, where in read cloud sequencing we use high molecular weight DNA and partition these long fragments of DNA within microfluidic droplets. Each droplet is tagged with a droplet-specific molecular barcode that is 16 base pairs long, and the library preparation reactions are done within each droplet so that by the end, all the reads that come from the same droplet are tagged with the same molecular barcode. And by doing this, we can computationally deconvolute which reads must have come from the same original physical long fragment of DNA. Read cloud sequencing was originally developed in the context of human genomics as a way to phase haplotypes, but our group has recently been able to develop this technology for use in a metagenomic context to sequence mixtures of organisms. The methodology was recently published in Nature Biotech last month, and this talk will focus on a clinical application of the technology. But if you're interested in more of the technical details, definitely check out the paper. So in this study, we applied metagenomic read cloud sequencing to examine the microbiome of a patient undergoing hematopoietic cell transplantation, which is a medical procedure often used to treat patients who have blood-related cancers or other disorders. And this patient population is super interesting to study from a microbiome perspective because, unlike healthy people, they are administered many different antibiotics within a short period of time. And they often observe intestinal domination, where their gut is dominated by a single or a few species. And within patients going through this treatment, lower diversity in the microbiome has been found to be associated with poorer outcomes and higher mortality. So one of the goals of the study was to be able to use this new technology to see if we could find genomic clues as to how certain species come to dominate over others within the gut, within this patient population. So given the high degree of inter-individual heterogeneity, we chose to focus our study on a time course of multiple stool samples collected from a single patient. So here is a graphical representation of their microbiome across 5 different time points, where each color represents a different species of bacteria or organism. And you'll see that at time point A, which represents the pre-transplant time point, their microbiome starts out looking very diverse, many different organisms, but then the diversity decreases over time over the course of treatment, and by time points C and D, we get this intense domination of E. coli. And so one of the questions we had is, can we explain how this E. coli came to dominate? And so here we see where our samples fall within the clinical context of our patient. So this is our patient across time, and on the left we see a list of all the antibiotics this patient was on and when they were on them, as well as when they got GVHD and bacteremia. So GVHD is one of the major complications of hematopoietic cell transplantation, and bacteremia is when there is a bacterial infection that makes it to the bloodstream. And so another question that we had is that given that we knew the bacteremia was an E. coli bacteremia, was the E. coli that infected the bloodstream the same strain of E. coli that was dominating the patient's intestine? So here is an overview of our workflow. We had 5 stool samples as well as the bloodstream isolate, the E. coli that caused the bloodstream infection. And for our stool samples, we performed both read cloud sequencing with our 10x Chromium library prep, as well as conventional sequencing using a TruSeq Illumina library prep. And then we generated draft genomes for each organism within each sample. So for our read cloud samples, we used our Athena assembler, and then for the conventional samples, we used MegaHit, which is a conventional short read assembler. And then we binned the resulting assembled sequences into various bins representing each organism. So by the end of our workflow, we had draft genomes for each organism present within each sample. And this allowed us to do some comparative genomic analysis between stool samples at different time points, as well as between the E. coli strains present in the gut and the one that was causing the bloodstream infection. So first, I will present some technological results. This table shows a list of the organisms for which we were able to produce a high-quality draft genome using read cloud sequencing for time point A, where high quality means more than 90% completeness and less than 10% contamination. So using conventional sequencing and assembly, that was only able to yield 7 high-quality genomes. So we see a technological advantage where read clouds are able to assemble a greater number of high-quality organism genomes from the mixture. So now if we look at how well we were able to assemble E. coli specifically, here is a Circos plot showing the draft genomes for E. coli for time point C in the outer track and time point D in the inner track. So each track shows both a blue and a dark gray assembly, where the blue assembly is from the read cloud sequencing and the Athena assembler, and the dark gray assembly is from the conventional sequencing with a MegaHit assembler. So if we look at the assemblies, it's clear that the Athena assemblies shown in blue are much more contiguous, whereas the gray assemblies are much more fragmented. And to quantify this difference, we can use a measure called N50, which is a measure of contiguity. And for our Athena assemblies, we see about a 10-fold improvement in N50 for both time points. And so this highlights the ability of read cloud sequencing to generate much more complete and contiguous genomes. Another major advantage for read cloud sequencing is that we found that it was better able to detect antibiotic resistance genes within the metagenome. So what we did was align the metagenomes that were assembled using Athena against the Comprehensive Antibiotic Resistance Database called CARD. And by doing so, we were able to detect the presence of resistance genes within the microbiome across the various time points. So the number of resistance genes that we were able to detect are shown below. And an important note is that there was a difference in the number of genes we detected and the number of unique genes we detected. And that was because some antibiotic resistance genes were present in multiple copies in the metagenome. So for example, if we look at time point A, there were 7 copies of this gene called TET-O and 5 copies of this gene called CFXA3 and 3 copies of MEF-A and so on. Whereas if we looked at our conventional assemblies, these genes were only assembled in 1 copy because of this ambiguity that we talked about earlier, where the assembler has trouble placing repetitive sequences within the correct positions in the genome. So the ability to detect antibiotic resistance genes if they occur multiple times across the same organism or different organisms in a microbiome, is one major technological advantage of read cloud sequencing that has major clinical utility. So now I'm going to switch gears a little bit away from technological results and focus more on some of the biological insights that we were able to obtain. So the first is kind of getting at that question of whether the E. coli strain that was dominating the intestine was the same strain that caused the bloodstream infection. And to get at this question, we performed pairwise alignment of the E. coli draft genomes from the various time points and between the intestine and the bloodstream isolate. So here in the table, we see for each pair the total bases that were aligned, the average percent identity, and the total number of SNPs between the two genomes. The bottom row is an unrelated strain that we got off of NCBI to use as a comparison. And so we see that based on the very high average percent identity, more than 99.9%, and the relatively low number of SNPs, that the E. coli strain that was present in the patient's gut before the transplant was very likely the same strain that persisted through the period of domination and also eventually caused the patient's bloodstream infection. The two graphs on the right are showing basically the same finding in graphical form. So the way to read these plots, called synteny dot plots, is that one genome is on the x-axis and one genome is on the y-axis, and there is a line plotted for every position where the two genomes are identical. And so if we were comparing two identical genomes, then we would expect a perfect diagonal line going from the bottom left corner all the way to the top right corner. And that's basically what we see here when we're comparing the intestinal strain from time point A to time point D, as well as comparing the bloodstream isolate to the intestinal strain at time point C. And what we don't see are any inversions or deletions or duplications of a large size that would suggest that the strains are different. And so we thought this was a really interesting finding, that it was the exact same strain that was present before treatment in the patient's microbiome. Granted, it was present at a very low abundance, like less than 2% of the reads during that time point were E. coli, but it was the same strain that went on to eventually dominate the gut and cause the bloodstream infection. So our next question was, is there anything that could explain what made this E. coli strain special? And indeed, so if we go back to time point A and look at all the various organisms that were present in the patient's microbiome, we looked at the number of antibiotic resistance genes that each of these organisms had in their genome. And during time point A, there were 87 resistance genes, and of those, 46 belonged to E. coli. The rest of the resistance genes were widely dispersed across many other organisms. So the organisms with the next highest number of genes were classified to Eubacterium and Lacnospiraceae, and each of those only had 5 genes. And so these results support a model in which our E. coli strain was able to achieve intestinal domination as a result of the extreme selective pressure due to the antibiotics the patient was on. Whereas all the other organisms were wiped out by the antibiotics, the E. coli strain was highly resistant and was able to flourish in all the niches left behind. Here is a table showing the types of resistance genes that were present in the pre-transplant E. coli strain. So we see that a majority of the genes encode proteins related to drug efflux pumps. And there were also genes that were related to resistance to aminoglycosides, polymyxin, and bacitracin antibiotics. Additionally, there was an extended spectrum beta-lactamase, CTXM27. And in fact, CTXM type enzymes are responsible for most of the extended spectrum beta-lactamase E. coli infections in the US. Overall, this case study highlights how advanced DNA sequencing technology can help elucidate complex biological phenomena occurring within real patients. We conclude that read cloud sequencing and the Athena assembler are better able to generate more contiguous draft genomes for individual organisms, as well as place repeated sequences in the proper genomic context. Furthermore, we showed that the microbiome can be a reservoir of highly resistant organisms, which can come to dominate the intestine under the extreme selective pressures during hematopoietic cell transplantation. I would like to close by thanking all the members of the Bot Lab without whom this project would not have been possible. If you have any questions, I would love to hear from you at Joyce_Kang at hms.harvard.edu. Thank you.